So this is a video about transcription. So here are some of the underlying principles of protein synthesis. The idea that the order of bases in DNA determines the order of amino acids, that's through the triplet code. So the order of amino acids determines the tertiary structure. That'll be due to where the positioning of the hydrogen, the ionic and the disulfide bonds holding that specific tertiary structure in place. So linking those together, you can determine that the order of bases in a gene determines the function of the protein made, because different tertiary structures of proteins give proteins different functions. Here's protein synthesis, so just a quick diagram. So we've got double-stranded DNA here. During transcription, you make a molecule of pre-mRNA that contains exons, which will be expressed, but introns that aren't expressed and need to be spliced out. So splicing occurs to remove introns, leaving mature mRNA. That mature mRNA will then travel out of the through the nuclear pore and arrive at a ribosome, where translation occurs and the polypeptide is produced. That polypeptide will then be folded into a specific tertiary structure, depending on where the hydrogen, ionic and disulfide bonds form. So we're going to start going through the process of transcription. It's a long answer question, so you need to know all the steps in detail. In addition, you need to know the differences and compare and contrast between this and DNA replication. There are a lot of similarities and differences. So we start off with a section of DNA that's unzipped by DNA helicase. So hydrogen bonds that did exist between the complementary bases have been broken. So difference to DNA replication, it's only a section of DNA, not the whole DNA molecule that's been unzipped, that the hydrogen bonds have been broken, but it is DNA helicase in both. In transcription, only one strand acts as a template. In DNA replication, it would be two. We do have complementary base pairing, but we've got RNA nucleotides which are attracted to their complementary bases. So we've got A with U, C with G. We've also got A with T, where the DNA base nucleotide was a T. So the attraction, we've got the hydrogen bonds that naturally form between the complementary bases, holding those nucleotides in place. RNA polymerase will then come along and we'll join these nucleotides together, so joining the sugar phosphate backbone. So joining the nucleotides together with phosphodiester bonds. What's produced at the end of that process is pre-mRNA, which still contains introns that would need to be removed. So splicing occurs to remove the introns. You produce a molecule of mature mRNA, and that can travel through the nuclear pore and head towards a ribosome to be translated into a polypeptide. In reality, the mRNA would be a lot longer, but it's hard to draw in a picture. So this is a typical long answer question that you could get asked, and this is a typical mark scheme that will go with it. So linking to the pictures that we drew and the process, you need to know step by step, but also similarities and differences to DNA replication. So just thinking about that question, what if you're having to compare and contrast? They both use DNA helicase, both have hydrogen bonds broken by DNA helicase. The difference would be that DNA helicase has two strands that act as a template, both do, whereas transcription is just one strand. Another difference would be that it's DNA nucleotides in DNA replication, not RNA nucleotides, but they are both attracted due to complementary base pairing. They both do involve polymerase enzymes to join nucleotides together, but in transcription it's RNA polymerase. In DNA replication it would be DNA polymerase.